So, good morning, everyone. My name is Christian Winsberger, Medical University of Vienna, and now we'll try to show you how you can use neuroimaging in combination with transcranial ultrasound stimulation. Now, some disclaimers. I am a shareholder in a medtech company, advisory board member, and I do have some patents on all kind of stuff. Right. I'm going to give you first a very, very, very brief introduction into tFUS, into transcranial ultrasound. Then I'm going to spend much more time on the tooth setup, on transducer designs, how to use neuronavigation in this respect, and what kind of workflow you can use in your experiments. I'm going to spend a little bit more time on a very cool option that you have in ultrasound for target validation, and that is using RF, so acoustic radiation force imaging. Now, ultrasound, as the name suggests, is using sound waves. Well, in principle, we've got two different kinds of waves. First of all, we have longitudinal waves, typical sound waves, right? So you push something back and forth, like you have a loudspeaker that goes back and forth, and that causes compression of the air. And these waves are then traveling and are carrying the sound information. And we also have transverse waves, right? But this is something that we won't see in air. We won't see this in water unless you go to the top of the water, like on the sea, where you see the waves going up and down. For ultrasound in the brain, it is mostly longitudinal waves. I should mention here also that we have shear waves that are also caused by longitudinal waves that have some sort of transversal part in there, but most dominantly it's longitudinal waves. Just one slide on the mechanism of tFUS. Well, in principle, what tFUS waves are doing is they are causing very small displacements in the tissue that we are sonicating, right? These displacements are in the order of micrometers, so very, very small displacements. But, as you will see later on, these displacements can be measured using MRI. There are three mechanisms that are proposed for the effects that we see in ultrasound, in transcranial ultrasound stimulation: thermal, mechanical, and cavitational effects in there. Well, number one, the thermal effect. This is what's being used in high-intensity focused ultrasound, so in ablation techniques, where you use ultrasound to get energy into the tissue on with a very high specificity, spatial specificity in there. And this is used in ablation techniques to heat up the tissue and degenerate the tissue there. And this is what you probably all heard about in tremor treatment, where by ablating two very small parts in the thalamus in there, this helps patients suffering from tremor. In low-intensity focused ultrasounds, we are not using thermal effects at all. In fact, we are really trying to keep the temperature rises at the focal spot below something of a degree centigrade. The main mechanism we're using in low-intensity focused ultrasound are mechanical changes. So the exact mechanism is a little bit, it's not really clear, but because of the displacement that occurs from the ultrasound, the membranes are changing, membrane functionality changes. We have some gating effects in the membranes, and these cause the modulatory effects. Finally, cavitation. So cavitation means that the sound waves cause little air bubbles to pair in the tissue in there. This is an effect that's been used in ultrasound variants that are aiming at, for instance, opening the blood-brain barrier, but also in low-intensity focused ultrasounds, which is the focus of my talk here. This is not happening. Yeah, as I said, so the low intensity tFUS causes changes in membrane dynamics and modulation in the ion channels in there as well. It is important to remember that whether you have an excitatory or inhibitory effect in low intensity focused ultrasound very much depends on the neural state the tissue is in. And as I said, the precise mechanism is somewhat unclear. This is the tFUS setup. So this is a classical setup if you are using a low intensity focal, also high intensity, but if you use focal ultrasound in an experimental setting. So what we have here, let's just open up the laser pointer. Yeah. So we have a driving system. And this driving system is consisting of a signal generator. So this is what's producing the ultrasound frequency. So typically we use something between 200 kilohertz up to 1 megahertz. This is generated here, the signal generator. Then you put it into an amplifier. And from the amplifier, it goes into a matching networks to reduce reflections on the cables in there, and then it enters this transducer. The important part of the transducer, the really important part of this transducer, is this piezo layer, right? And this piezo layer is actually the one thing that really causes the ultrasound waves to occur. So this is a special material. When you put electrical voltage on it, then it will simply change its size. Right? And by using the amplified signal, ultrasound signal, you then have also a change in this piezoelectrical element in there, and this will produce the ultrasound waves that you're using. Importantly, we need to make sure that this ultrasound transducer is nicely coupled onto the tissue we would like to sonicate. And this is important as sound waves tend to be reflected when you have changes in the acoustic properties in there. So you would always use some kind of ultrasound gel to make sure that the coupling between your transducer and the actual tissue, the skull that you want to sonicate is fine. Now let's have a closer look at these transducers and how the design of the transducer actually affects focus sizes in there. So this is our transducer. Again, this is the one thing that goes back and forth, or in this way, left and right. Now, there are two main parameters in transducer design, and this is the diameter, the aperture of your transducer, and it's the curvature of your transducer. So these two parameters determine the focus size or the focus shape that you have in there. Now, if we use a smaller transducer in there, then you can see we have less nice definition in the longitudinal direction, so in the Z direction. If we have a smaller curvature, so we have more coverage in there, then we can go from this more cigar-like shape into a more really spherical focus that we can achieve. And if we, with changing the frequency, we are changing the wavelength. So if we decrease the frequency, we are increasing the wavelength, and for this reason, your focus size will become bigger. So changing or determining, selecting, choosing your transducer geometry has very strong effects on the focus sizes and the focus shapes that you will be able to generate. Now, these were transducers that were just using a single element. So there's one thing that's vibrating, that's causing the ultrasound to be generated. You have the option in ultrasound or in transcranial ultrasound to use so-called multi-element transducers. Multi-element transducers, as the name suggests, are transducers with multiple elements in there. And these multiple elements, these individual elements, are driven individually. So you can choose phases of the input signal to these elements, and with this you can change the focus. You can change the focus size and the focus position. So a simple approach is transducers that have these concentric individual transducer elements in there, and if you use such an approach, you can very nicely change the depth of your focus. So you can really change where in the brain you would like to you would like to have the tFUS effect to occur. This is very helpful in many applications, because not only can you now target different areas in the brain, it actually is a very, very elegant way of having a sham control in your experiment. Keep in mind, all kind of brain stimulation techniques are really benefiting from sham control, so you can exclude any effects that are simply caused by the fact that a stimulator is present and is doing something. If you use multi-element transducers, you can have a more or less the perfect sham control. You're just changing some of the stimulation parameters in there. Your participant will be unaware of any changes of those, and you can have a look at the difference in the effects in two different conditions. Once where you're actually stimulating the one part of the brain you're interested in, and in the sham condition, you just change a few phases in your driving electronics in there, and then you're stimulating something completely different without the participant knowing anything about that. So it's really, and you have the same side effects. You have the same noise, the same impressions on the participants. So that's really quite cool. You can go further than that and not only have these concentric transducers in there. So this is an example for a 256 matrix transducer. So this is a transducer as we are using it in the Citrus project, which we'll see a little later on. So there are 256 elements in there, and you have 256 individual channels that you drive individually. So you can change phases and amplitudes in these different channels. And with this, you can really very nicely change not only the depth at which you are stimulating, you can actually also vary the angle in there. So these are so-called steerable transducers. And they are very, very beneficial because they are, in this case, you are able to reach areas in the brain, your target regions, not only by sort of changing the inclination of the transducer on the head, but by simply changing the stimulation parameters in there, right? So these are steerable transducers in there. Now, from the transducers to neuronavigation. Neuronavigation is a very cool technique. It allows you to know where you are in space, I would say. It depends on trackers. The typical trackers that we're using look a little bit like that. So this is one tracker, and each tracker has 3 of these small spherical elements that are just reflective. And your neuronavigation system consists of a camera and 2 illuminators in there. So the illuminators will send out light. This light is going to be reflected by these tracker elements in there, and it's going back to 2 cameras in there. And because the cameras, like your eyes, have a spatial distance in there, so they see the reflected spheres on a slightly different angle, the system can calculate the position of the tracker in space. This is a very robust technique. It works really nicely. A standard camera like this allows you to track a number of trackers simultaneously. So this is really working really nicely. So then you sort of need to put the trackers on your participant. These are 2 MR-compatible approaches, or things that work in the MR quite nicely. You just take a piece of tape and tape it on the forehead of your subjects in there. That works quite nicely. There are other options where you can put it on the mouthpiece, which is more nicely then connected to your participant, right? Because here in this example, it's on the skin, and therefore, if the skin moves over the skull, you also have these movements in there. Just a quick slide on the accuracy of this. So this is work by Michael Wohletz, who had the task of finding out how accurate are these neuronavigation approaches really, if you're interested in distances between different trackers in there. And he developed a very nice approach, I think, for doing that. So this is a board that contains a number of 18 passive spheres. So these are the exact spheres, these rich reflective spheres that you saw on the trackers before. What the system is doing is it's using these 18 trackers, these 18 spheres, to form 54 trackers on one board simultaneously. And then you can use this, show it to the neuronavigation system, and from the 54 trackers that are defined there, so each tracker contains 3 spheres, you can actually calculate around 1,500 distances between the individual trackers, and you can very nicely then measure the accuracy in there. This is the comparison across the field of view in there. The important thing is down here. So the accuracy of this system is really very nice. So it's about 0.5, 0.6 millimeter in there. So neuronavigation works really nicely. This is the short message of that. Now, if you do this in the scanner, things are getting a bit more complicated because you have to make sure that whatever you use in the MR scanner room is actually really MR compatible in there. So you are using the camera, neuronavigation camera up here. You have some MR compatible device where you can actually mount the whole thing in there, and then you just put it close to your scanner in there. Right. Let's talk about what level of accuracy are we actually required to meet for these applications in there. Now, thinking about transcranial electrical stimulation, well, typical focus sizes are here in the order of centimeter or so. Focus location is quite variable depending on where you put the electrodes in there. If you take a look at TMS, transcranial magnetic stimulation, focus size is around a centimeter or so, and your focus is on a cortical area. Now, for ultrasound, the focus size can be a lot smaller. It's typically in the millimeter range. So depending on the configuration of your transducer, you can have focus sizes of 5 millimeter or even less. Typically, and that's one of the main advantages of ultrasound, you can go very deep into the brain. So you can target very deep brain nuclei in there. And this, as a consequence of this, very small inaccuracies of your transducer positioning will actually have quite severe consequences on your target location in there. And just to give you a nice example of that, or hopefully a nice example of that. This is the brain. This is the transducer that we put it. And if we zoom into that, let's assume this is our target, the target we'd like to hit. Now, if we now just slightly tilt the transducer on the outside, because the target is several centimeters away, typically 7, 8 centimeters away from the transducer, this small change in the angle will actually shift our target a centimeter or even more away from where we want it to have. So for that reason, it is really important that you have very good control on where you put the transducer and what are the angles, the inclinations of the transducer when you put it onto the skull. Now, typical workflow, we will have a planning session. Then we will do some simulations in there, and then do the actual stimulation session. Well, simulations, why do we need to simulate? We need to simulate how the sound is actually traveling through the skull and the brain and hitting the target. And why do we have to do this? Well, because we are all different. So these are 3 CT scans of 3 individuals. And to show you the differences a little bit nicer, I've just zoomed into that, and you can see that the skull thickness varies quite significant across these different individuals, right? Now, the thickness of the skull is really an important factor when it comes to something that's called phase aberration. What is phase aberration? Well, let's consider we have 3, let's call them ultrasonic waves that are traveling around here, and they're all in phase. Right? So in phase means that the maxima and the minima of the 3 different waves are in the same position there. Right? So if we now assume this is the bone, this is the skull, and you see different thicknesses of the skulls in there, and that will cause a shift in the phase. So the 3 different ultrasonic waves that we had before, that were in phase before, will now no longer be in phase. Right? And if they're no longer in phase, they will have a different interference pattern, and your focus will change, the spatial position of your focus will change, and actually the focus layout will change very, very strongly, right? So for that reason, we need to simulate the aberrations that occur and use this information beforehand to actually calculate where our focus will be and what's the brain area we're going to stimulate. So for the planning session, we typically acquire anatomical data. We acquire something that we call a pseudo-CT. This is just a way of getting the images I showed you before of the skull, just to get an estimate of skull thickness and skull anatomy in there. We might want to acquire a functional localizer. As mentioned before, we might want to acquire diffusion, DTI data in there, and also some resting state data that helps us to actually find the perfect target for the experiment we're interested in. And we use this for target definition, for transducer position setting, and for the sonication simulation, right? Now, for target definition, we need to have the anatomical data and any localizers, functional DTI or resting state localizers we might want to have. In order to get the best transducer position, right? You need to be sure that to be able to put the transducer there, we need the anatomical data and the CT. And for the sonication simulation, again, we need anatomy and the pseudo-CT data in there. Now, then you need to simulate the sonication effects. There are a number of software packages available. This is an example from software called K-Plan, where you are placing the transducer on your favorite spot outside the skull, and then it calculates you the location of the focus in there. And now, depending on whether you have a steerable transducer or not, you can now keep the position of the transducer constant and use the steerable options that you have to move around the focus of your ultrasonic stimulation. So if you're not using MR for actual stimulation session, then you would need to register your participant, right? So that the neuronavigation system knows where your subject is. You will try to position the transducer exactly as possible as in the planning session, and then run the experiment, right? Now, if you have MR available in the stimulation session, you again need to register the participant in there. You also try to position it as good as possible. And then you put the whole setup into the scanner. Right? So, which means you have to have a MR-compatible tFUS system as well. But if you can do that, you acquire MRI data of participant, and then you actually run the real experiment. So if you're interested in doing online tFUS, where you acquire fMRI data or resting state data, whatever, at the same time with doing your sonication, this will be the approach that you can use for that. Now, if you can also do the following, so that's all the same. You can use inborn neuronavigation to validate the transducer position, right? So in this case, you would need to have a setup where the neuronavigation camera, the system is actually in the scanner room and is able to look at your participant plus seeing all the trackers in there to really validate where you are. Otherwise, you are in a sort of a fire and forget position. So you're positioning your participant, your transducer, everything is fine. And then you put your participant into the MR scanner, and then you just need to hope that nothing will move on the way in, and your participant will be very nice and not move at all, so that everything is static and everything is exactly the way you thought it should be when you did your simulations in there. Using inborn neuronavigation actually allows you to validate for that and maybe also change some of the parameters once it's in there, right? Right. So, how can we validate the target position in there, in vivo? So normally we would do the simulations, and as I said, we would hope that everything is done in a perfectly fine matter. So for that reason, the target will be where it should be, where we thought it to be in the simulations there. But can we actually validate it? Well, quick answer is yes, we can. Now, in high-intensity focused ultrasound, this is, for instance, the application in tremor patients. There, it's rather easy to do that because we are heating up the tissue, and MR is actually a very nice technique to measure tissue temperature. So we can use MR techniques, so-called proton resonance frequency shift approaches in there to measure the distribution of temperature in the brain. And if ultrasound, high-intensity focused ultrasound is heating up parts of the brain there, we will very nicely see it there. For the low-intensity focused ultrasound, we are not intending any temperature increase, right? So even our safety limits dictate us that we have to have very, very little temperature increase at the focus site. So here we can use a method which is called MRI, MR, acoustic radiation force imaging. And what we are doing here is we're using phase encoding gradients to change the MR signal in the displaced regions. Now, me being an MR physicist by training, I'm always trying to sneak in some MR physics into whatever presentations I'm giving. So apologies for that, but yeah, we're using so-called field gradients in there. The one and only thing you need to understand in order to understand most of the contrast mechanisms we have in MRI is that there's a very simple relationship between the spins that are rotating around and the magnetic field. The higher the magnetic field, the faster they will spin around, right? So in this example, if the magnetic field is the same everywhere, they will spin with the same frequency. If we change that, they will change in their frequency. That's all, right? With this approach, you can understand frequency encoding, phase encoding, all kind of stuff. Now, in phase encoding, what we are using is we are taking a bunch of spins in a perfectly coherent manner, and then we add a gradient. So we change the magnetic field depending on spatial position in there. And if we do this, we actually cause a phase change, right? So now the phase encodes for the spatial position in there. So this is what we call phase encoding in MRI. This is the approach that we use for actually getting images from the MR signal in there, right? Now, if we do this, so we're using a gradient to change the phase. We call this a dephasing gradient, right? Because the spins that have actually are all coherent are now no longer coherent. If we take the same gradient but in the inverse direction, we can compensate for that perfectly. Now in tFUS, there is one thing. In tFUS, what we do is we move some of the spins just a few microns in one direction. Right? So let's have a look what happens to the phases if we use tFUS. So again, we have this dephasing effect in there, right? And now we're applying tFUS here, which means that these spins are now moved a slight amount, right? They are moved, the spatial position has changed. So when we now take the rephasing gradient, so it's exactly the same gradient as before, just the opposite direction, we actually end up with a phase shift in the spins, depending on the displacement in there. So with this approach, we can, in a very nice, I think very elegant way, we can very nicely measure displacements on a range of micrometers using MRI. And this is so-called RF, acoustic radiation force imaging. Here are some examples how you can do this. This is in a phantom, so the cross section, so that's transversal to the actual sonication direction, and this is in the sagittal plane in there. And here are some examples for if you change how much sonication you do, you actually very nicely see a change in the displacement in there. So for an actual stimulation session with MRI, you do the same thing. You register your participant, you position the transducer, move everything into the scanner, you use inboard neuronavigation, but then you can do RFI. You have a short RFI scan. This will tell you whether your sonication is actually sonicating the right spot in the brain. And if you have a steerable transducer, you can now change the focus to the point you would like to stimulate. Right? And that's a huge difference. It really makes a huge difference because positioning the transducer in there and keeping everything static and having a non-moving participant in there is not as easy as it sounds. Right? So even if you have slight changes in there, you can now perfectly compensate for that, for readjusting some stimulation parameters. You do another MRI feed to validate that you're really there, and then you run your experiment. Right? So this is an example of such a setup in there. This is what we use in the Citrus project, where we are developing such a setup that includes a dedicated MRI coil. So this is the white thing that you see there. In addition, we have 2 transducers. So these are the 2 steerable transducers in there. And then we have a bunch of trackers for neuronavigation. So you put the subject in there, position everything in a nice way, and then you run your experiment in there. And I'm looking at the organizer. Do I have 4 minutes for a quick video on that? So just that you see very nicely how this really looks like in reality. So let me just move it over here. Right, so I'm going to skip that. This is just putting the stuff there. So this is the setup. This is the registration of the system with respect to the neuronavigation. Here you have the neuronavigation camera. You had it here. This is the brave subject in there with a mouth tracker. And this is how this subject is then being registered. Right? You put the gel onto the transducer. You add the gel pad on top of that, right? And then things are ready to be placed on the skull, right? Mm-hmm. Then it goes into the scanner. Yeah, another bit of ultrasound gel on top there. Then you put it all in there. Then you put the subject in, right? You can see plenty of trackers in there to make sure that the transducers are on the correct position, that the subject is in the correct position, that you control as much as possible of your experiment in there, right? And then you do your acquisitions in there. I'm gonna go forward. This is the simulation part. In the simulation part, you select your targets, the best positioning of your trackers. You do a simulation of that, and then you can actually see the sonication field that's being caused by that. Let's hop into there, right? So that's the sonication in there, and then just reposition the participant again, as exactly as possible in matching the conditions of your planning session. I can tell you this is really a challenge in there to trying to do that, to put the subject in there in exactly the same position, yeah, you need to try this a few times to be able to do that. Based on the neuronavigation that you're using on the scanner, in the scanner here, you actually know exactly where you need to be, and then you try to match the conditions as good as possible, right? And then you use this information and run the actual simulation, the actual sonication in the scanner there, right? Good, so let me go back. Right, and that's my final slide. So I'd like to acknowledge all the partners in the Citrus project, the funding that we received for that, and obviously all collaborators and all people in my lab. And thanks very much.